Hello, good afternoon, everyone. Thank you for joining our webinar, Environmental Health, presented by Environmental Health Language Collaborative. The mission of our collaborative, just to remind you, is to advance integrative environmental health research by promoting access, use, and harmonization of data through interoperable terminologies and best practices. You can become involved with ELK using the visiting our website, and also you could check ELK resource catalog. And if you want to contact us, you can email myself or Charles Smith directly, or contact elk@icf.com. You can also join the ELK email listserv. And you are welcome to suggest use cases and semantic needs for ELK to address, propose new activities, training, or event topics and speakers, and also useful resources that we could add to the portal. So with that, I would like to introduce our presenter for today. Dr. Bjoern Peters is a professor in the Center for Vaccine Innovation and the Center for Cancer Immunotherapy and the La Jolla Institute for Immunology. Dr. Peters began his academic training in physics at the University of Hamburg, where he graduated summa cum laude before earning a PhD in theoretical biophysics from Humboldt University in Berlin, also with highest honors. He then transitioned into bioinformatics and immunology through postdoctoral training at Humboldt University and later at the La Jolla Institute, where he has built a distinguished career spanning computational biology, immunology, vaccine research, and cancer immunotherapy. For more than 2 decades, Dr. Peters has been a leader in applying quantitative and computational approaches to understand adaptive immune responses. His pioneering work has advanced our ability to predict T-cell epitopes, enabling a deeper understanding of how the immune system recognizes pathogens and tumors. He has also played a central role in the development of the Immune Epitope database, or IEDB, one of the most widely used resources in immunology, leading its bioinformatics efforts since its inception. Beyond his scientific discoveries, and the most relevant to the goal of ELK, Dr. Peters has been instrumental in developing community standards and biomedical ontologies that facilitate data sharing and interoperability across the research ecosystem. His research combines clinical, experimental, and computational approaches to address important challenges in infectious diseases, allergy, vaccine development, and cancer immunotherapy. Please join me in welcoming Dr. Bjoern Peters. And I will stop sharing, and Bjoern, please take it away.
And I'm hoping I'm sharing now.
Yes.
You can see my screen, perfect.
Yes.
Yeah, I want to really thank you for the invite. I think we met like on a cross NIH meeting that was about like data interoperability, and I was asked to present here. And so obviously NIEHS is not where I normally present, so I'm hoping I'm going to give enough of a background of kind of like where my work is coming from, and hopefully how to connect it to the work that you're doing. And I want to address ultimately how we use ontologies to make our work easier and better, and that is motivated through the work on the Immunotherapy database. As you just heard, my background is very different. I got like sucked into this field as a necessity of getting the kind of adaptive immune response data modeling done in a good way. This is not like my training or something like that, but I'm at this point a keen advocate of the use of ontologies as data standards for knowledge representation. So as I said, the key project that really has been getting me into this is the Immunotherapy database, which is why I joined LJI in 2003 originally. This has been just funded again, so we are at this point 25 years of funding, which is exceptional for an NIH-funded resource. So Alex Sette used to be my PI. Now he's leading the IEDB together with me. He's the card-carrying immunologist. I'm more the computational bioinformatics guy. The goal of the IEDB is to provide a freely available resource to catalog experimentally derived immune epitopes in a number of contexts: infectious disease, allergens, autoimmune diseases, transplantation, and then actually use that catalog of data to create tools to predict new epitopes and identify like potentially immunogenic reactions and stuff. I'm not going to talk about that. Here I'm going to talk about the database itself. So overall, I'm going to make arguments about like why what we are doing is, I'm just going to assume that what we're doing is good, and that is based on kind of the success of the IEDB, as I mentioned, was refunded, and that is because at this point we have managed to curate 26,000 papers, over 7 million individual experiments, and it is really a new resource that is also reflected in, at this point, we are approaching 35,000. I think at this point we are above 35,000 citations to the resource, and the website traffic. I thought when we hit the pandemic, we had this huge bump of getting over 1,000 visitors a day. And this has been keeping on going up. It's kind of amazing the kind of engagement you can get from users if you do something consistently over time. So we are doing some things right. So what is it that we're doing? So we have this literature curation process. So we're taking papers that come out from PubMed that are about epitopes, and we find the ones that are about epitopes by first doing keyword searches that are then going through essentially natural language processing classifiers that are identifying which abstracts are most likely relevant, which are then manually classified into really relevant or not, and then read by curators and manually curated. And we mean that. So we have a team of PhD level curators that read the papers and enter the data from the papers, which might be figures, might be materials and methods, free text, and put it in a structured format. They are PhD level curators. We started with undergrads, and that didn't go well at all. It ends up reading papers and really understanding what's going on there requires a high level of expertise. So this data structure now obviously is meant to enable something that you can't do by just putting papers side by side, namely to compare it and able to accumulate information that you have from different sources. So this is when you go to iedb.org, and I can just repeat it again. This is a free resource. You can go there right now, and you're going to get the same kind of homepage. Actually, it might be slightly updated right now, but you can perform queries here looking for immune epitopes. Again, immune epitopes are the molecular structures in antigen that are directly recognized by the adaptive immune system, so antibodies or T-cell receptors. So, so one of the applications would be you can go in here and say, I'm looking for any kind of epitopes, so immune system targets that are recognized in Pseudomonas aeruginosa. I should have picked something else that I can actually pronounce. And you type this in, it auto-completes, and you click find, and now you're getting all of our data back. That data is organized in terms of these are the 422 epitopes. Many epitopes are linear peptide sequences. And sorry, the data is organized in different tabs that are essentially different views of the exact same data. So there's 68 antigens, so this would be proteins in which these epitopes are contained. Then there are the specific assays in which they're measured, and this could be T-cell assays, B-cell assays, or MHC ligand assays. Then there's the specific immune receptor, so there's like these are 5 antibodies that are in their sequence that are recognizing specific epitopes in Pseudomonas. And these are the 94 papers from which that data is derived. So if nothing else, and if you're caring about immunology at all and you're dealing in this space, I would very strongly encourage you to just go to the IEDB, put in the kind of organism that you're interested in, and see what's out there, what has been characterized, and then at least these papers should be highly relevant reading material, typically. So going a little bit into more detail, here you have now on the assay tab one specific assay, and assays are really the atomic unit that we are capturing in the IEDB. So you can always click on the numbers on the left here, and then you're getting the information back, which is essentially like, you can imagine this is essentially a database. You have like field value pairs, and you have like the information about the paper it was published in, about the epitope itself, like where is it in the protein, and then you have information about where is that information found, like in table 1 of this paper. And then this was like a epitope recognized actually by a mouse that had a transgenic TCR injected in it. So this is not a regular mouse, but it's a transgenic mouse. And then you get the information about the assay that is being run. So in this case, they did a proliferation assay on T cells from this mouse that showed a direct ex vivo response that was class 2 restricted to this peptide. And there you go. Importantly here, for the purpose of this talk, we are putting on the left the typical things that a user would expect, namely the normal words like spleen or H2 class 2 or so. But on the right, we have pearls that are linking to the definition of each of these terms in ontologies. So it looks like they're free text, and users can interact with them as free text, and you can query as free text with autocomplete and all these things, but they're linking back to ontologies, and that allows us to do, if actually we started by originally people just typing in text, and then obviously you have all the typos you can imagine, and you have like unclarity about like what labels to use, etc. The ontologies enforce precise terms. They allow for using synonyms, and they allow for hierarchical queries. So for example, and these are, we always use OBO ontologies, and I'm going to go into details about that. But those are linked to common like knowledge bases like the NCBI taxonomy. So for example, when we say Pseudomonas aeruginosa, that is in the NCBI taxonomy. It is actually an ancient one, 287. That's one of the early entries in the taxonomy, which then allows you to query also for, say, all bacteria, which will include this, or there's different strains which are children of this. So it's not just the label, but you have a hierarchical structure that defines the term that we're using here in context. So I showed you right now all this cool data that we have on Pseudomonas aeruginosa, and that, if you don't know, is one of these pathogens that is of increased interest because there's a lot of antibiotic resistant strains. So there's lots of interest in this as like an important pathogen that actually has high clinical relevance in a setting where you're dealing with immunocompromised patients. So then I'm demonstrating here how, by not going to just ask for that the organism, the epitope source is Pseudomonas, but we will also ask for that the host is actually human. I showed you transgenic mouse recognition, right? That is very cool if you care about the specific TCR in that transgenic mouse. And I mean, many clinicians will not care about mice at all. And then say, I showed you antibody and T cell recognition, but say you only care about T cells. So you can now ask more precisely. When I asking for Pseudomonas epitopes, I actually mean those recognized in humans and those recognized by T cells. and then you can rerun your query, and you're going to get results back that is now much fewer epitopes. Obviously, because you're kind of like filtering down. There's only 7 proteins left. There were 68 before, and this is not because the IEDB captures negative data as well. This is not just because all the data was not reported because it was tested and missed. That nobody has tested anything apart from these 7 antigens, as far as we are aware, in Pseudomonas for T-cell recognition in humans, which is a small fraction, given that this is an important pathogen. This looks completely different when you look at, say, TB. And then you can be even more specific and say, well, we don't just want humans, we want humans who actually are infected. And then it ends up, there hasn't been a single study where humans that have been infected with Pseudomonas are tested for T-cell responses. All the data is from healthy individuals where you see these responses. So what I'm trying to show here is that by curating, not just, other resources will say, whatever, this is epitopes for Pseudomonas or something. Actually, there's no other epitope, but whatever, expression data sets or so. And by us trying to narrow down precisely how the experiment is performed that leads to the data that we have, that allows us to generate queries that allows the user to pick up what they actually care about, right? And it's not like that what I just showed about like whatever humans infected with Pseudomonas, maybe you don't care one bit. Maybe you want to know, for example, you might only care about antibodies. Or you say, no, I'm working in an animal model and I want to see all the mouse data. I don't care about the human data, right? There's no right or wrong. This is about precisely defining the data that we have in a way to then display it back to the user. So going a bit more into the IEDB data structure, we have the epitope itself, which often is a peptide. And then we have where do we have the information about it? Typically, in our case, this is a journal article, but there's also like user-based submissions. Then we have the epitope source, which is often the protein from which it's derived, but you can have, say, you have antibodies recognizing complexes of heterodimers, say, so you have more than one protein, say, that is recognized. And then you have the assay in which that epitope is tested. So that is an assay that must measure B-cell or T-cell responses on MHC ligands. And this assay is kind of the experiment, again, the key concept for us, which is preceded by like, why do you think that person, that sample that you're testing will show some kind of immune response? And that might be because people are infected, they're naturally exposed, they were vaccinated, etc. So for lots of these aspects here, there are existing ontologies, and they pre-existed before the IEDB. I mentioned the NCBI taxonomy before, that has been around for a long time, as has UniProt or the gene ontology. So we have, when we started with the IEDB, we tried to connect to this resource of ontologies. And the reason for that is because they already provide lots of value. If you're looking, say, at UniProt for a given protein entry, I'm showing here hemagglutinin in UniProt, that shows you not just that there is such a protein, but also from UniProt we can grab information like that the mature protein is actually spliced into a signal peptide, and then an HA1 chain and HA2 chain, which are separate, and then they have different than independent conformations. So there's lots of data like that out all over the place. And when we talk about making IEDB data interoperable, the goal is obviously for us to link to other people out there. We are not going to repeat the kinds of studies that have been done to characterize the structure and variation in hemagglutinin proteins when we talk about the immune recognition. Actually, with UniProt, we've had a nice collaboration to actually get the epitope data into the protein records themselves. So I mentioned before the NCBI organism finder. Here I don't know why actually all my examples here are on influenza apparently. You have this separation into not just influenza virus, which is a species, which is somewhat controversial for viruses, but then you have these subtypes, and then within the subtypes you have different sequences, and all this information again comes from massive amounts of studies done by much bigger consortia than the IEDB that are dealing with the, say, genomic variations of these different influenza strains. And again, by connecting to that, we're gaining from it. And then the NCBI taxonomy is actually linked to UniProt protein records, which then in turn link to these things like the whatever, where's the signal peptide in this, etc. So some of these data sets that we're using are not necessarily what we want to completely expose to our users. What I'm showing you here is the tree you have to click through in the NCBI taxonomy on the left and on the right to end up with Homo sapiens. And I believe it's like 40 different clicks where you would always have to know, oh, is human under teleostomy or whatever, or not. This is a really good hierarchy for the NCBI taxonomy, which is trying to deal with taxonomic information in every context. But for the typical immunologist, this is a level of detail that is actually not helpful. So here's an example how we want to connect to a resource that it provides really valuable information. At the same time, we do not want to have our users deal with the extra information that is useful in the context of that resource, but not for us. So here we have done this approach of essentially pruning down the taxonomy to those nodes that are relevant in immunology, and actually we tested for our users and random postdocs that are forced in the lab to ask essentially what do they understand. And now, I mean, compare it. I will challenge you if you think you can click through this without error in less than a minute. But this, everybody should know that whatever humans fall under primates and are vertebrates, etc. And this is short enough that you can actually get some useful information. So what I'm trying to say here is just because we are trying to tie to general useful vocabulary, we do not have to make our users suffer. We can provide pruned versions of this vocabulary that have the exact same semantic value like the full version, but that can happen in the background and it doesn't have to be in the user interface. The disease ontology is another thing that we are using. I'm showing here is the OID, which actually has done a massive amount of work in connecting to, as you will know, you probably maybe are familiar with like SNOMED or UMLS or MESH, or there's a whole bunch of disease vocabularies out there, and the disease ontology, apologies, is doing a great job of connecting them all. So by using disease ontology terms, we can then link out to the corresponding SNOMED term or ICD-10 code or whatever. And so in this case, this is peanut allergy. That is one of the, we're capturing allergic epitopes, so you can search for the disease of having peanut allergy. And then in the IEDB, as I showed you in our data model, we have the disease and we have the corresponding immunogen, or in the case of allergy, the allergen that is linked to the disease. And so here's an example where we work with the disease ontology to always, when we have it, for an infection, we'll have the pathogen, and for an allergy, we will have the allergen linked to it, and then we use that. So for example, when you have here occurrence of allergy to Timothy grass, in the IEDB, and then you fill out that the allergen is egg, then this would be flagged as an invalid entry. This is interoperability, right? Egg would be Gallus, NCBI taxon. The occurrence of allergy to Timothy grass has a certain DOID. And we have the link in the ontology that Timothy grass allergy is triggered by pollen from Phleum pratense, which is Timothy grass. And egg is not that. Chicken egg in this case is not that. You can have chicken egg allergy. So this would be an example where we can then use the linkages between the ontologies to provide consistency checks of the data entered in the IEDB. And the other way around, like you have people who are allergic to benzylpenicillin. And benzylpenicillin is a type of penicillin drug. So essentially, when you're allergic to this, you have an antibacterial drug allergy, and a drug allergy. So essentially, you can now search for, we put as a in the curation, we put in, you have allergy to benzylpenicillin, and you can search for, I'm looking for drug allergies, and then you get that back along with others. I'm hoping I'm making the point here that by using these external vocabularies that are interlinked, we can both improve our queries, we can gain knowledge. I mean, gain knowledge just means that users might start off with drug allergy, and we don't have to clarify for every entry that we put in, is that this is a type of drug allergy. We just put in the compound, and then it's inferring the kind of other information. So, sorry, I'm still cough recovering. A key other ontology that we're using is the ontology of biomedical investigation. That is actually what to us was missing when we started connecting to the OBO ontology world. So there was no description of actually the experiments that was given. So we have all kinds of different experiments cataloged, like ELISpot assays or ELISA or ICS or bioassays that measure IL-2 cytokine release. So as a user, you might want to see, like, where do they show an IL-2 response, and you might not care what the way it is measured, or you can go through the method and say, well, just give me anything I can measure with an ELISA or so, and then get that back. So as more resources represent information using these formal ontologies like the IEDB does, and there's a continuously growing set, especially OBO foundry ontologies, the more we can get interoperability because we are linking through these reuse terms. So you can say whatever, you can have this penicillin molecule, you can click from that going to other records in, say, CADI on what are the what is known about the molecule. You can jump from that to mass spec repositories where the molecule was found in samples, or to structural databases that have been shown how that interacts with other molecules or so. By doing this, we are essentially enabling queries across resources. This doesn't necessarily have to happen all in the IEDB, right? You might be getting an answer from an IEDB query, and then you're getting your identifiers that allows you to translate to a query to a different resource that you're working with. And here, yeah, so one of the things that we had been looking into that, like, we can now check out all the things that cause allergic reactions and ask for, can we use kind of like the chemical scoring methods to distinguish the types of compounds that you have allergies, that the people have been reported to have allergies to, versus where there's not, which is, to our minds, there's no other resources like that out there. And then compare what are the chemical similarities or differences between these compounds. So I mentioned the ontology for biomedical investigations, which was kind of the glue ontology for us to interconnect the different ontologies that were out there. This was established in 2007, along with the OBO foundry itself, actually. And it was really a, do I have that in the next slide? I don't think I have. So there was a number of similar projects in different kind of experimental domains. So there was like the MJet people and the proteomics people and the functional genomics people or so, and they all worked on different aspects of describing experiments, and they were kind of focused on their technology, but it ended up there were so many things shared, specifically when you're talking about, say, sample preparation for an experiment. You want to indicate that that same sample is then used, say, for a transcriptomic and a proteomics experiment. So that led to this joining of effort. This has been going on for 20 years now, with weekly phone calls and somewhat yearly, it's sometimes more, sometimes less, face-to-face meetings. This is an open project. This is key. All of the OBO Foundry ontologies are open. If you want to join and contribute, I can definitely speak for OBI, which I'm heavily involved with. We would be thrilled. We love to have more people helping. These are volunteer efforts, and we can always require more. We can always use more hands-ons. So this is how OBI looks like in the context of other existing OBO ontologies. So at the high level, you have in green here BFO, and I would ask you to ignore that. And then you have in blue the kind of OB terms, things like specimens or assays or human subject enrollment. And then this ties to existing OBO ontologies, such as I mentioned KBB4 for different molecules. I mentioned the NCBI taxonomy, but also things like the biological processes in Go, which might be things like downregulation of interferon alpha response or something that is talking about what happens in biology versus the planned processes are the kinds of things that we are doing as part of an investigation, such as performing an assay. So we import these Foundry ontologies and try to connect to all of them. And again, like a little bit of history here. I'm sorry. The gene ontology. I'm sure most people on this call are familiar with its existence. Not everybody knows the whole history. The gene ontology was kind of the first data interoperability project in biology, or the first massive one at least, maybe the PDB as well, where people from different model organisms came together and said, like, we are doing worm-based, and we're doing fly-based, and we're doing Drosophila, Arabidopsis, and we're doing mice, etc. And all of these were at the bloom of the whole cross-organism genomics research, doing experiments where they were kind of attacking and saying, this gene looks like, when you knock it out, we have the following phenotype, and we want to annotate these things with the phenotype. And that's what we formed the Gene Ontology Consortium for, which then got all these people together and agreed on like a structured vocabulary to describe what is the function of genes in terms of what is their molecular function, what biological process do they contribute to, what cellular components are they expressed in, or are their products are found. And this is, at this point, I just looked it up, 48,000 citations. That's pretty awesome. So if there's a successful project, it's this one. And it had like two killer apps. The first one was the goal of the gene ontology was to say, okay, we learned this in worm, we learned this in Arabidopsis. What could this mean in human? So the goal of the whole thing was to say, okay, it looks like, because these genes are, they have a sequence homology, and then you find a human gene, and you say it's homologous to this one in worm and this one in mouse or whatever, and in there it has been shown to do X and Y, and then you want to kind of like align these annotations, and that was the goal here of the gene ontology. compare sequences of genes with known function, with new ones, and then have the same vocabulary that allows you to then transfer likely function of newly discovered genes. And this is obviously definitely at the start of the Human Genome Project. This was this was the thing. And then Killer App 2 came later. So by doing this well, there came the whole concept of gene set enrichment analysis. And this came up with microarray experiments where people measure gene expression across the entire transcriptome, hopefully, and ask like which ones the genes are up and which ones are down, what is their shared function, what is their shared locus of expression, and then identify essentially what is actually happening here. And that then helps you to identify common features of, say, pathological gene expression patterns. So that was Go, and around Go was this network of biomedical ontologies that I really want to apologize. Sorry. So things like, say, the cell ontology. When you want to say, okay, this gene, I don't want to only say like what is the cellular compartment it is a gene product is found, but also like which cell types is it found in. So that was a natural expansion. And then there were others like that. The sequence ontology, which annotates all kinds of sequence variations or so. So it kind of, there was this nucleus around the Go that then added to more and more ontologies. And then other people like us with the IEDB came in as well, saying like, we really want to use your vocabulary, but we need kind of more than what you're covering. So that led to these, I mentioned this, transcriptomics, proteomics, metabolomics, but then all of these were kind of using Go somewhat differently. So rather than having all of these things done side by side, we wanted to merge these efforts and come up with, because this actually recapitulated like the different, like whatever, worm base, fly base, and mouse genomics, whatever, were just as incompatible before, so we want to be better than that. So that then led to the OBO Foundry, where the idea was, okay, let's try to make sure that the vocabularies that we create are going to be interoperable. And the idea was to form a family of ontologies that are following a core set of principles that everybody voluntarily adheres to. And then the idea is that people contribute to their own ontology and help others and connect with others with the goal of having a set of interoperable ontologies at the end. And then these principles, we're trying to make sure that we can work with these ontologies together. And there's simple things, they're actually really simple, like for example, agreeing on this, it must be in this following machine readable syntax. There were competing syntaxes out there, and essentially we said like it has to be either OBO format or OWL format in a certain way, and then there's a way how to round trip them, so that's fine. There's naming conventions, there's licensing issues, they have not gone away, but they're also really important for reuse of other people's projects. And then the idea was to have this family that would be non-overlapping, and that is actually probably the most challenging one. So if you're working on an ontology and we're interested in participating, you can submit it to the OBO Foundry for peer review and then get feedback on how you're doing. This is the original member ontologies, and then what was added to it. And then this is how it looks like. This is biological process, one example of the component of the gene ontology. And then you have things like, yeah, you should always be able to contact people with questions, etc. And for term requests, this kind of information that I'm showing here on this page. is like required for each of the ontologies to provide essentially the community with enough information to contribute to the ontology or get feedback on it. I mentioned a number of the principles that we had. The first ones I would consider common sense, such as, I mean, there should be licenses that are compatible, etc. There should be versioning as you update things. You can't just toss the old. Then there's these metadata principles, which are there. I mean, it's common sense to have some, but it's not clear how they should be formulated. There's certain conventions that you actually by sticking to certain conventions, it makes it easier to read ontologies when you're skipping from one to the other. And then there's the very foundry-specific principles, such as the delineated content, that different ontologies should ideally not overlap. Also the concept of collaborations, that you shouldn't start something that just like encroaches on someone else's territory, but rather contribute to. So in 2014, we started, like, there was the original paper that described these principles, and they were well thought out, but they were not necessarily always clear how to action them. So also, as with any kind of journal review, you have a problem finding reviewers who have the necessary expertise to perform reviews, and they don't have infinite time to do something that is an unpaid volunteer activity. So as much as possible, we wanted to try to automate this review process and try to take the principles that we had and reformulate them in a way that we can directly test this, like, does your ontology comply with this, yes or no? And I think I'm low on time, so I'll skip through some of these things, but like, one is this open principle, where the original textual formulation of the ontology must have an open license that allows for reuse. Now, what does that mean? So first of all, what does have a license mean? So we said, okay, you must have in your OWL file a property that says DC license equal, and then point it out. And then we said, okay, we are not going to go through these endless debates of what is an open license. We're going to ask you to use one of two, which I believe is CC BY and MIT license. I forgot. So essentially, and if you want to argue, you're welcome. But this is a testable thing. You must have the license as part of your file, and we had agreed upon these two licenses, which actually covered everything that we had. And then you must, in the OBO registry, which is the file that I just showed, that must match what you have in there. And so then this is the OBO dashboard. So this is, at this point, this is the first run I think we did in 2020, where we then just said, okay, this is the ontologies, and for each of these principles, we're trying to compute, are you hitting it or not? So here, for example, Kebi is not, did not have the ontology license, so it gets like a red X. And what ends up is that this works. Ontology developers, this is kind of like a gamification. You're seeing how well your ontology behaves to others. And then this is a good way to just make people fix things and make the red X's go away. So it's a way how we can, within the OBO foundry, enforce principles. Well, it's less force. It's actually people will voluntarily do things. Now, the biggest problem, or one of the big problems, is the whole like interoperability and overlap, which is very hard to test for. So Bill Baumgartner and Larry Hunter run reasoning over pairs of ontologies. So that asks for the logical consistency of pairs of ontologies, and that revealed that most of the ontologies were extremely had zero conflicts, but the problem was that they were completely talking past each other. If you have two ontologies that share no logical, no classes, no relationships, it is impossible to have any kind of conflict between them. but we knew there were conflicts. So one of them is like that the cell term was defined in both the gene ontology and the cell ontology, and they were not compatible when you read closely. We, in order to make those things apparent, but this was like not, you couldn't see that because that was just a textual label on the term cell, but you didn't know that they were talking about the same thing. So the BFO ontology should have taken care of this by providing an upper ontology layer and saying, oh, they're talking about the same kind of cell here and not just, I don't know, cell in the spirit of, I don't know, cell automaton games or a jail cell or what other kind of cells you could imagine. But BFO actually didn't do that job. So we've been working on providing a different type of upper level ontology, which builds on BFO, but gets a little bit more explicit. So BFO will stop at a material entity, and we are trying to dig more down into biomedical ontology. So we will have classes in COB for things like organism that then makes sure when we are now going to test for that each OBO foundry ontology is going to be placed under COB classes, which is then that's going to be a testable thing, and that's going to allow us to check for overlap and interoperability between these classes. So that was the last slide I had. Sorry if at the end this was getting a little rushed maybe. I hope I convinced you that using ontologies in the IEDB, which again, we started primarily for our own organization of the curation effort, led to an enhancement of the kind of queries we can do within the IEDB webpage itself, but also connecting to others. Utilizing the OBO ontologies has given us kind of like an outside check on what we're doing, enforcing stringency, which sometimes means that we have to do some extra work but in the end it does pay off quite often, specifically when you're getting into areas that, I mean, for me, I can say this, like where I don't know much about, say, we just had like spatial transcriptomics. I know nothing about that. And I've learned a lot talking about the domain experts when we were trying to model that kind of data. Contributing to OBO ontologies is easy. This is a common concern I hear when people say, well, I looked it up, but there is no ontology that does X, or I can't find my term so-and-so, so I'm just going to do it all myself. That is, in my mind, the wrong answer. The right answer would be, okay, if you're lacking something and you see an ontology where it would fit in, please contribute back to it. I see Jennifer Fostel is on the call, so she's been doing that for NIEHS projects for a while as well. And it's more painful than just making up your own little spreadsheet of terms, but you are benefiting in the end from connecting to other things, getting more consistency in the vocabulary that you use yourself, and actually connecting the data you annotate towards these terms to other resources that are out there that use it. And as I said, like OBO ontologies should be responsive to user requests. As with every volunteer project, it's not always as perfect as it could be. But and also obviously I want people to keep in mind when you're comparing OBO ontology to whatever, SNOMED or something like that, where there's actually massive amount of money support in the background. There's certain, not everything can be done from a community effort, but I have seen in the last 20 years now of OBO work, we have seen so many other efforts come and go. The best selling point for OBO is that all these volunteers are still there and are still working on it and are still putting the effort in, because this is actually useful, in contrast to a large number of efforts that come and go with like an initial large amount of enthusiasm and no staying power. I do want to acknowledge here, primarily here, the IEDB folks, as the IEDB was the driving use case for much of what I presented. We have other projects as well, and I specifically want to highlight Randy Vita, Sebastian Dusing, and James Overton that we are working with on the ontology work. And with that, I will stop and cough for a while while maybe somebody asks a question.
Thank you very much, Dr. Peters. I don't see any questions right away in the Q&A. And people can also, just to remind our audience, you can raise your hand if you want to ask a question audibly rather than put it in writing. But you addressed I can start with a question. You addressed situation when you don't have a term, and rather than creating your own small ontology, go to the relevant ontology and request to include the term. What if you have an opposite situation when you find your seemingly similar term in multiple ontologies? How do you choose which one to use?
Yeah, so the first one would be like, what are those ontologies? I would go down to the OBO family of ontologies, which should be narrowing it down. If you look for, say, NCBI Bioportal, you will have an infinite number of sources for the same terms, many of which are copied over. NCBI Bioportal has NCBO Bioportal is really useful for getting like a very, very, very broad overview. But I mean, there's lots of student projects in there that haven't been touched for forever. I would very much recommend never using it as a source of terms, unless as a very last resort. So I would always start somewhere else, look for the higher quality. And when I say higher quality, I think this is somewhat objective. Again, like a student project, ontology is not going to help you, right? Nobody's going to add anything. Nobody's going to change anything when you need a sibling term or whatever. And it's nobody's going to deal with maintenance, transfer, etc. So when I say quality, you can argue about quality in some ways, but I think everybody would agree that maintenance is a key consideration, and that is one of the things that is also captured in the OBO Foundry. But there's others as well, right? Like and then I mean, okay, I have my own personal preferences. Like there's other things that also have been around for the longest time, like NCI thesaurus or mesh subject headings, which are not really ontologies, and they don't claim to be ontologies, but people use them as if they were, and that can be just fine if you have no other way to go. But if you want to do the kinds of things like I showed in terms of inferring knowledge or so, that gets hard, to say the least, because the kind of relationships within these to other ontologies are not clearly defined.
Thank you. I see there is a question by Ivan Bolton. What challenges might an environmental or exposomic-based resource face in attempting to add new terms? Maybe a bit general, even though do you want to come off mute? Are you do you have in mind a specific ontology?
So I think you're asking Evan, right? If I can answer it. So there is an exposure ontology that we have been trying to work with. The concept of exposure, but that is actually, I don't actually know what the status is. For the IEDB data that we're doing, we have been working a lot with the kind of administered exposures. So things like different routes of vaccination, etc. But I believe when you're saying environmental exposures, it's more like the kind of thing, whatever, you inhale fumes at the gas station. Not something that is targeted and intended, but something that happens. And to some extent, we have something somewhat similar for infectious agents, where you say you live in a malaria endemic area. That doesn't mean you have malaria, but it means that your likelihood of getting exposed to Plasmodium falciparum is drastically higher than if you live in San Diego. So I think all of the kind of challenges that are there, I don't know what exposomic necessarily is, but if this is like kind of different routes of exposure to essentially material entities and tracking down like what types of exposures there are, like the administered versus the accidental or environmental, there's definitely lots of people are working on that, and I don't think that that's a fundamental concern. If you're specifically interested, please email me, and I would be happy to follow up if you're not already involved with the. Sorry if I'm trying to remember now. Who was in charge of that? It was like exposure ontology or something. I forgot what it was called.
I want to say it's Anderson. Here's a question, another question from John Graydale. What tools are available to support OBO Foundry ontologies, speaking particularly of finding, mapping, annotating with those resources?
Yeah, I mean, for finding, I think the best tool that's out there is OLS, which is hosted by EBI. As in, like if you're putting a term in there or so, like finding it, getting it displayed in a hierarchical format, and also like limiting what ontologies you want to get results back from. It works quite well, really well as well. Actually, I believe a bioportal, you can also do those things, but I think that the defaults are harder to limit it to OBO Foundry, if that's what you care about. In terms of mapping and annotating, I don't believe there's any OBO Foundry specific tools out there. Obviously, there's a lot of ontology tools for mapping and annotating in general. Yeah, and many of them are using, or obviously most of them would be allowing for that, but I don't believe that there's specific tools. It would be cool to have more specific tools, but yeah, I don't think there are.
Yeah. All right. I think there are no any further questions, and we are on top of the hour. So thank you so much, Dr. Peters. And I hope to see our audience at the next webinar, ELK webinar, which hasn't been scheduled yet. The announcement will come. Thank you very much. Have a great weekend, everyone.
Thank you for the invite.